Okay, welcome to week 2. The topic for this week is DNA sequencing. So, last week we have seen the structure of DNA, we have seen how DNA replicates, how DNA is expressed into proteins. So, we have touched all the basic topics in the first lecture, and this week we are going to learn about DNA sequencing. So, first of all, before we learn how DNA sequencing is done, it is important to know what DNA sequence means. So DNA sequence basically means the order of the nitrogenous bases, the order of A, T, C and the G's. So basically, as you know, DNA is a polymer and it is made of nucleotides. And if you recall, a nucleotide has three components. It has a pentose sugar, it has a nitrogenous base, and it has a phosphate group. So pentose sugar and phosphate group is same in every nucleotide. So what makes one nucleotide different from the other? It is the nitrogenous base which makes one nucleotide different from the other. So the order of these nitrogenous bases is the sequence of DNA, or it is a DNA sequence. So how one gene is different from the other gene? It is only on the basis of the differences in the order of these nitrogenous bases. So the order of nitrogenous bases is the DNA sequence. So how DNA sequence is done? It is done by this technique. It's called dideoxy chain termination sequencing method, developed by Sanger. So this is the most common method which is used for DNA sequencing. But currently, so this is the method basically on the basis of which DNA sequencing is done. But now currently we have a lot of modifications of this method by which this sequencing has become very efficient and very fast, and full genomes can be sequenced in 1 or 2 days. I mean, it has become such a efficient technique now. But the basic technique is what I'm going to explain. It's called dideoxy chain termination sequencing method, developed by Sanger. So before we actually learn the technique, we need to know the term what is dideoxy. So as we discussed in the last lecture, we talked about the nucleotide. So the structure of nucleotide is in front of you. The nucleotide in DNA is called deoxynucleotide. So in DNA, if you recall, this is your pentose sugar, this is your carbon number 1 carbon, this is your 1' carbon, this is 2' carbon, this is 3' carbon, here is 4', and on 4' is attached the 5' on which this phosphate group is attached. So here is your nitrogenous A, T, C or G in DNA. At carbon number 2 in DNA, because DNA is deoxy, so there is H. If you recall, in RNA it is OH here, and on 3' it is always OH group. Whether it is DNA or RNA, at 3' end there is always going to be OH, and on 5' is the phosphate group. Now what is dideoxynucleotide? Dideoxynucleotide is that nucleotide, or it's a modified nucleotide, in which on the 3' carbon, instead of OH there is H. So which means that there is one oxygen less here. The second oxygen which is less is on the 3' carbon. So that is why it is called dideoxy. So which means that on the 2' as well as on the 3' there is H group. So if you see RNA, there is OH here, but in DNA there is H here and OH here. But in dideoxynucleotide there is H at both the 2' and the 3'. That is why it's called dideoxynucleotide. So the technique I'm going to teach you by which DNA sequencing is done, it involves both kinds of nucleotides, deoxynucleotides as well as dideoxynucleotides. The point to remember here is at 3' carbon in dideoxy there is a H group, and in deoxynucleotide there is a OH group. So after having this information, now I'm going to move towards the technique. I'm going to explain you how DNA sequencing is done, and then I'm going to talk to you what are the modification. So this is the basic technique I'm going to discuss right now. So how the current method of DNA sequencing is different from these. So first of all, we are going to learn about the conventional method. So basically, before we study the technique, we need to understand in little more detail how DNA replicates itself. So DNA replication, as I discussed in last week's lecture, that DNA, as you know, it's double stranded. The two strands of DNA, they separate, and both strands act as parent strands, and whatever is the sequence on each DNA strand, that acts as a template, and on that template new daughter strands are going to be formed, and the daughter strands which are going to be formed, they are on the basis of complementary base pairing rules. So it's basically simple. Two strands, because DNA is double stranded, the two strands separate, both strands act as parent strands, and on the basis of the sequence in the parent strands, new daughter strands are going to be formed on the basis of complementary base pairing rules. But this technique, this is a procedure, this is a mechanism going on in our living cells. It is not that simple. There is the involvement of some enzymes and few more other things which I'm going to tell you. So first of all, the formation of a daughter strand, which is formed on the basis of complementary base pairing rules, takes place with the help of an enzyme called DNA polymerase. Now DNA polymerase is an enzyme which adds the nucleotides, complementary nucleotides, and makes a new daughter strand. But this DNA polymerase cannot elongate, this DNA polymerase cannot initiate this process. So this DNA polymerase needs the presence of a primer. Now what is a primer? You can see here, this is a primer. So the primer is a short stretch of DNA or RNA. So remember that primer is a short stretch of DNA or RNA. In our living cells, the primer is made of RNA. Okay, so this is a short stretch of DNA here. So in the labs, when we do the techniques, the primer is made of DNA. So it's a short stretch of DNA, and it is, remember that always two strands of DNA, they are antiparallel in nature. If this is the template strand and this is the new strand going to be formed, this new strand will be complementary to this, but their ends are going to be antiparallel. So if this is the 3' end of this strand, it will be 5' here. If it is 5' here, it is going to be 3' on the opposite. So this is the primer. It's a short stretch of DNA. Okay, so the requirement of is a primer, and it needs a free 3' end. So on a deoxynucleotide, on the 3' end is a OH group. So a DNA polymerase needs a hydroxyl group, free hydroxyl group at the 3' end. If you remember, as I explained you in the first lecture, how a covalent bond or how a phosphodiester bond is formed between two nucleotides. It is formed between 3' carbon of one nucleotide and the 5' carbon of the second nucleotide. Suppose this is your nucleotide number 1, this is your nucleotide number 2. This is the 3' end, 3' carbon. On 3' carbon there is a OH group, and this is the 5' carbon. On the 5' carbon is the phosphate group. So the 3' carbon having the OH group and 5' carbon having the phosphate group, they're together going to lose one molecule of water, and that is going to result in the formation of a covalent bond between these two, and that will link that those two nucleotides. Coming back to this, so the requirement of a DNA polymerase is a hydroxyl group at the 3' carbon. So if this is the last nucleotide here and it has this group here, the DNA polymerase can add the next nucleotide, and that nucleotide, the next nucleotide, can be linked to this nucleotide by the dehydration reaction, they together lose one molecule of water that will link them. So the requirement of DNA polymerase is a hydroxyl group at the 3' carbon. So this is the basic information we need to know how DNA replicates. Now let's see what is going on in this kind of a technique by which DNA sequence can be, we can find out what is the sequence of DNA. And remember that in this kind of a technique, we are trying to find the sequence of this strand of DNA. And we are going to find the sequence of on this strand of DNA by using this technique. So in this technique, what is done is, this technique involves these two kinds of nucleotides, deoxynucleotide and dideoxynucleotide. And remember that the main difference between deoxynucleotide and dideoxynucleotide is, in deoxynucleotide there is a OH group, and in dideoxynucleotide there is a H group. So in this kind of a technique, basically template strand will be taken, and then in this, so basically we want to know what is the sequence of DNA. The two strands of DNA will separate, and then a primer is going to be attached, which means in this reaction primer has to be attached, has to be provided, and DNA polymerase has to be provided. So these are all, here are written all the ingredients in this reaction. So the DNA, double-stranded DNA will be there, primers will be there, DNA polymerase will be there. There will be both kinds of nucleotides, deoxynucleotides and dideoxynucleotides. So these are the deoxynucleotides, and remember that deoxynucleotides are of, there will be of 4 kinds, because here the base can be A, T, C or G. And similarly, dideoxynucleotides can be of 4 kinds, because whatever is attached here, if it is adenine, it will become dideoxyadenine. If it is thymine, it becomes dideoxythymine. So basically, what I am explaining right now is a conventional method. In the conventional method, what it was done, people used to take 4 tubes, and in the 4 tubes they used to take DNA which they want to sequence. They used to add a primer, they used to add the enzyme, they used to add all these 4 these deoxynucleotides, and so they had 4 tubes. In one tube they used to add dideoxyadenosine triphosphate, which means the nitrogenous base is adenine. In second tube everything will be same, but they will take the dideoxythymine. In third tube they will take dideoxyguanosine triphosphate, and the fourth tube they will take the dideoxycytosine. And also these dideoxys, they were radio labeled, means they were labeled with the radioactive isotopes. They emit radiation, and those radiations, they can be detected on the X-ray films. So they used to have 4 tubes, and in 4 tubes they used to do like that. Let's see, let's study what happens in this tube, in which the dideoxy is with adenine. So you can see here, this is the strand, the DNA is double-stranded, 2 strands will separate and the primer will attach. Now the primer has a 3' OH group here. Suppose here is G. So this DNA polymerase is going to add here C. If it is A here, it is going to add here T. If it is T here, it is going to add A. Now remember that in this tube there is dideoxy ATP and it is radio labeled, but there is also normal deoxy ATP also. There are both things. There is a dideoxy ATP and there is a deoxy ATP also. So both things are there. So but this dideoxy is in small amounts. So there is a chance, this is in small amount, this is in more amount. There is a chance when there is a T here, here A has to be attached. Maybe it can come from here or maybe it can come from here. It's all random. Suppose this dideoxy ATP gets attached here. So if dideoxy gets attached here, after this reaction will stop. Now why this reaction will stop? Coming back to here, remember that dideoxynucleotide has a H group and DNA polymerase needs OH group. Dideoxys don't have OH group, they have H group. So that is why, remember that very important point, wherever dideoxynucleotide is going to be incorporated in this reaction on this strand, the reaction is going to stop. So here if there is incorporation of dideoxy ATP, this reaction will terminate here. Okay, and remember that in one tube there is not only one molecule of DNA, there are multiple copies of DNA. Maybe this is one molecule of DNA. In this reaction will stop here, but this is another molecule of DNA. In another molecule of DNA, maybe where here was A, the incorporation was of the normal ATP, so the reaction will proceed. Next is C, G will be incorporated. Again now there is T. Now again there is T, there is a chance dideoxy can get incorporated. If dideoxy gets incorporated here, this reaction won't proceed. And this is another molecule of DNA. Maybe here is your T, normal A. If this is G, this is C, then this is third T here. Maybe now dideoxy is incorporated, so the reaction will stop here. So you can imagine this is one tube which has multiple molecules of DNA, and in different molecules different things can happen. But in one tube, when these different things are happening, the result of this kind of reactions will be, there will be formation of DNA fragments of different sizes. So because you can see here this only this much DNA fragment is formed. This is smaller, this is longer than this is longer than this. So which means that this is the conventional method in which people used to use dideoxys, which were radio labeled. They used to have 4 tubes. In every tube they used to add a different dideoxys, and in every tube these kind of reactions were taking place, and because of that different sized fragments were being formed. And what they were doing, let me go here because we have a better picture here. Yeah, see these are your 4 tubes. Okay, so this is the tube with the dideoxy T, dideoxy C, dideoxy G, dideoxy A. Now in these tubes these reactions have taken place, and in this fragments of different size, then they used to run these fragments which are in every tube on the DNA gel. Now DNA gel, how does it works? Actually there is one lab at the end of this course, basically on the 7th week, where you will be also doing electrophoresis. Now what is electrophoresis? Electrophoresis basically means the separation of DNA by applying electric current. So you can so usually what we do, we run the DNA gels. So DNA gels are made by a jelly kind of a material called agarose. Agarose is a kind of a very porous material. There are pores inside, and the DNA runs through those pores. If the size of DNA fragment is long, it's going to run slow. So suppose, and in these kind of gels there are some wells. Wells are like small compartments where you can load your DNA samples. So the sample from this tube can go in this well, the sample from this can go here, the sample can go here, the sample can go here. Then you apply the electric current. Okay, so the fragments is in this tube are going to run with the current. The fragments which are longer in size, bigger in size, they will move slow, so they will be more towards the well. The fragments which are very small, they will move away from the well. Same thing here, same thing here, same thing here. The fragments which are bigger in size, they will stay more closer to the well, and the fragments which are small, they will move away. So they used to run the gel, and on the basis of the size of the different fragments, this gel used to run. Suppose in this, like we have studied with A. So as you remember, there was a formation of three different kinds of fragments here. If you go back here, see this. So this one is the shortest, this is in between, this is the longest. So this fragment in the gel is going to be somewhere here. Maybe this is the fragment, okay, and the intermediate will be here, the shortest one will be here. So the sample from all the 4 tubes you can run on the gel, and then because the dideoxys are radio labeled, then it can be analyzed on the X-ray film. In the X-ray film you can see these kind of bands, because wherever these fragments are, they do have the dideoxys, and dideoxys are radio labeled. You will see these kind of bands, and on the basis of the size of these fragments, the sequence of DNA can be deciphered. So basically, which means that if this is the tube with A where you have added dideoxys, it means if this is the basically A here, if this is the size of if this is the tube with A, it means basically here is T, and here is also T, here is also T, because dideoxy was attaching to the T's because it is complementary. But remember that there are 2 strands of DNA. So if it is T on that strand, on the opposite strand it is going to be A. So it will be noted down as A here, and A here. So this is the way, and also the data from the other tubes will tell you there is a G here, there is a G here, there is a G here, because this reaction is only going to stop when G is going to go and complementary bind with C. So this DNA sequence they tell from the opposite strand, and the opposite strand is complementary. So on the basis of the pattern of the fragments you get in those reactions, you separate those fragments on this gel, and then that is transferred on this X-ray film, and then whatever is the size of those fragments, that actually tell you what is the sequence of DNA. So this is the conventional method how it used to be done. So for this kind of a reaction, people used to work in 4 tubes, and they used to work with radioactivity, which is little dangerous, and it was a slow process. Now what scientists they have done, so this is the pattern of these DNA fragments. What they have done now, they have, instead of using radioactivity, they use fluorescent dyes, and fluorescent dyes can be of different colors. So like here you can see this dideoxy ATP is labeled with red fluorescent dye, C is labeled with blue, G with green, and thymine with yellow. So these are fluorescent dyes. So, and they are of different colors. So which means that there is no need to use 4 different tubes, because these colors are different. So you can do these reactions in one single tube. So wherever C is there, it will give blue color. Wherever yellow is there, it will give you wherever T is there, it will give you yellow color. Wherever A is there, it will give you red color. So on the basis of using these fluorescent dyes, there is no need to use 4 different tubes. The reactions can be done in one single tube, and the differentiation can be on the basis of the colors. And this is the kind of data you get. You get the peaks with the different colors, and those peaks tell you which nitrogenous base is there. So wherever there is a red, this nitrogen base, if this is blue, this is this. It's all on the basis of what you have done in your reactions. So this is the scientist, his name is Fred Sanger from Cambridge. He was the one who developed this method, and he was also given the Nobel Prize for finding this method. because it has a very big application. So this is all what I talked. This is the old method which I just discussed with you. So the terminators, so the dideoxys, they are the terminators, because wherever there will be incorporation of the dideoxy, the reaction will stop, because the dideoxys have H at the 3' carbon instead of OH, and DNA polymerase needs OH group. So this is just like a picture to show you what is actually going on. So this is the DNA strand you want to sequence. The two strands is separate, and the primer is going to bind here, and in the DNA sequencing technique the primer is provided, and it is a DNA primer. The replication, how does it takes place inside our cells? It is basically the same. The two strands is separate, and then there is an attachment of a primer, but in our cells the primer is RNA, because there is an enzyme called primase. It attaches the RNA primer, and then after that it's the same thing. So this will keep on elongating, this strand will keep on building up if the deoxy nucleotides are being attached, because deoxynucleotides, they have a OH group at the 3' end. As soon as the attachment of dideoxy is there, this chain reaction will stop. This reaction will stop. So this is all what I discussed. So next, each reaction mixture from the 4 tubes is electrophoresed in separate lanes at high voltage on a polyacrylamide gel. Pattern of bands in each of these 4 lanes is visualized on X-ray film. Location of bands in each of the 4 lanes indicate the size of the fragment terminating with respective radio labeled dideoxy nucleotide. DNA sequence is deduced from the pattern of bands in the 4 lanes. So this is what I just explained, and I have also posted 3 videos. Please watch those videos. They will really help you in understanding what I'm talking to you right now. Okay, so this is all the old method. So if this is the tube with G dideoxys, so wherever the side, you can see if you're getting this fragment, it means at this point there was one G. So remember that dideoxy G is going to complementary attach to cytosine, but on the opposite strand it is G. So the sequence is for that basically. So currently in the labs we don't use it was a slow procedure, but now it's an era of automated sequencing. In automatic sequencing, as I said, everything is done in one tube, and it uses fluorescent dyes, and on the basis of colors these nucleotides, they can be distinguished. And also these days the labs where the DNA sequencing is done, they're not done manually these days, they're done by robots. Okay, so robots are operating, they are automated, and they can do all the things. They can like pick the bacterial colonies, they can make the DNA from them, and then they can do all the steps further. So this is the era of automated DNA sequencing, which is very, very fast now. So this is all what I explained. This is the current method which is basically used. So everything is done in one tube, and nobody runs the gels these days. So basically all the sample which is coming from this reaction goes in one gel lane, or it is called a capillaries. So they are so thin. This is a DNA sequencer machine basically, and you can see with these machines the computer is attached in which you can get this data. So the reaction is done in one tube, and through these capillaries, it's called we don't use these conventional gels. These sample is passed through these capillaries, which are very, very small narrow tubes, and the sample passes here and it is exposed to the laser, and laser is going to activate the fluorescent dyes, and these fluorescent dyes, when they get activated, they are going to emit some radiation, and that can be detected by the detector, and it will give you the data in the form of these peaks which are of different colors, and it is called a chromatogram data. So the chromatogram data comes like that, and the different colored peaks tells you about the different nitrogenous bases. So all this data, so as you know, almost all the genomes of organisms are already sequenced. So human genome was sequenced maybe 15 years back. So what is the significance of doing all these sequencing? See, remember that central dogma: DNA codes for RNA, RNA codes for proteins, and proteins are the real players. They are doing all the functions in the cell. So this there are a lot of things involved in that. You can study different diseases, you can study basic things which are going, because there are a lot of things which we don't understand now. So all the data, these labs, these are all very specialized labs, because these sequencers and all these techniques, they're not present in normal labs. These kind of sequencers are quite expensive, so they are present in some very like in institutes. There are some central labs which have a lot of funding. They buy these big instruments, and then the whole institute can make use of that. So and there are some specialized institutes which are totally devoted to do the DNA sequencing of different organisms. So what is the purpose of why we should do this? Because we can study a lot of diseases, we can study like we can study the basis of a lot of diseases if we know the sequence. And all these DNA sequence which you get is deposited in public repositories. Basically there are different databases. I'm going to talk today about different databases. So all the information, all these sequences, they are deposited there, and the scientists from the whole world can make use of that data, that sequence data, and can do various things. They can find out how the protein is going to be there, what is it's going to be, the three-dimensional structure of the protein, making use of bioinformatics tools, various computer algorithms, various computer tools. You can if you have a DNA sequence, you can compare DNA sequences, you can find out homologies between different genes, you can make computer models, you can study where the protein is going to be there. You can do various things. So the data is deposited in these databases which I will be talking. So the analysis of DNA sequence is the basis of the field of bioinformatics. It is a field in which the biological data, which is in the form of DNA, RNA, and the protein sequence, to analyze the DNA sequence data with the help of computer algorithms and computer tools, makes this field which is called bioinformatics. Because by doing the analysis of this data, we can find out various things just on computer. So basically the field of bioinformatics has made the life of a biologist less complicated, because a lot of things which you actually do in the lab, you can now work on computer, and these are called all in silico studies. So on the computer, because in these databases, all the scientists which are working in different labs in this world, they deposit their data, and people can using these computer tools, using these computer algorithms, people can make use of the data from the other labs. They can compare their data with the other labs data, and can come up with different inferences. So that is what the field of bioinformatics is, to analyze the biological data, which can be the sequence of DNA or the sequence of RNA or the sequence of a protein, using the computer tools and various computer algorithms to solve various problems, to understand what is the structure of protein, what will happen if there is a mutation in the DNA, what kind of protein it's going to make. And you can create, getting this basic information, various drugs can be made. So it's a great field. We will slowly and slowly every week we will pick it up. So this is kind of you can see this is a kind of a robotic machine which is picking up. So as I said, it's all automated these days. DNA sequencing is not manual anymore. So this is a lab where DNA sequencing is done at a large scale. These are called high throughput technologies, where you can see this is a robotic arm, and it's picking up the colonies, the bacterial colonies, and it's going to make the DNA, and then the analysis. The analysis can be, after you make the DNA, you need to sequence it. You undergo that reaction, the chain termination reaction, and these are the capillaries which can have your samples. Those samples are then exposed to laser. The fluorescent dyes will get activated. They will give some radiation, and that can be detected, and the chromatograph data can be obtained. That is what is happening. So you can see here how many plates are there, and everything is going on large scale. So this is a mini prep robot. Means the robots can work at even a larger scale. So you can see here, this is a PCR machine. PCR is called a polymerase chain reaction, where you can amplify the DNA. If suppose you have very little DNA, you can actually amplify in the tubes. These are all PCR machines. And this is about 150 sequencers here. So these are all sequencing machines here, and these are the computers giving the data in the form of chromatographs. So these are all various these labs where all the genomics data is being produced. So genomics is a field where you study the genomes. The genome is basically in the form of DNA, and then DNA expresses into RNA. RNA makes the proteins. So this basically tells us how fast this field is going up. Most of these genome factories are dying out and being replaced by machines that can do massively parallel sequencing, 1.6 million well plates instead of 96 well or 384 well plates. So these are the current genomic technologies. So this is the DNA sequencer by Roche. This is equivalent to 10,000 old sequencers. This is another DNA sequence by Illumina. This is equivalent to 100 old sequencers. So this is a great field. It's growing very fast, and we hope it can solve a lot of problems. So as I have already explained, what is bioinformatics? So it's the interface of biology and computers, analysis of proteins, genes, and genomes using computer algorithms and computer databases. Genomics is the analysis of genomes. The tool of bioinformatics are used to make sense of billions of base pairs of DNA that are sequenced by genomics projects. So these are the three databases. Basically, GeneBank by NCBI, EMBL, which is by European Bioinformatics Institute, DDBJ, this is it's a Japanese. So we are going to use this. We are going to use GeneBank, which is at the NCBI website. And what is NCBI? It's a National Center for Biotechnology Information. So it's a US government funded resource, which is NCBI is a big database where the information about a lot of genes is there, a lot of research papers. I mean, there's a lot of literature which is called PubMed. So we will go on this side and we will study a lot of things. So these are some of the important species. The information is there on GeneBank. So this will be our main website where we will be doing our all analysis of the data. So National Center for Biotechnology Information is called NCBI. So this is the website, and NCBI is a resource. It's a US government resource for all the molecular biology studies. So you will see there, like here on the left side, there's a window. If you scroll down, you can go to Gene Bank, you can go to PubMed, you can go to various computer tools. And here is a window where you can put, so here is a BLAST, you can do structure studies, taxonomy studies. So we will be basically working on this resource, National Center for Biotechnology Information. This is American. Okay, after the DNA sequence is done, as I explained by the Sanger's method, the next job is to do the annotations. Basically, annotation is to find out, so there are two kinds of annotations. It can be structural, it can be functional. Basically, after the sequence we have found, so what is basically sequence? It is the order of these A, T, C, C's and G's. There's a long list of A, T, C, G's. So to make sense what is this A, T, C, G's, we need to do gene annotation. So basically we need to find structural annotation and functional. Structural annotation basically means to find out what are the different regions in DNA. So to find out, so I'm going to use few terms now, and I have provided in week 2 folder some extra reading material, so you can go over those material and you will be able to understand the concepts better. So there are different regions in DNA which are doing different functions. Like there is a promoter region in DNA. Now what is a promoter region? Promoter region is a region in DNA where an enzyme called RNA polymerase goes and binds, and it starts the process of transcription. So transcription is a process by which DNA converts into mRNA. So there's a promoter region. There are enhancer regions where some co-activators, they go and bind and they make the transcription faster. Then there are in the DNA itself, there's a promoter region where RNA polymerase goes and binds. There's a terminator region where the transcription will stop. Then there are regions called open reading frames. Now what is the open reading frame? Open reading frame are the regions in the genes which are protein coding regions, which code for the genes. So as you will go with this course, you will see that there is lot of DNA which is called junk DNA, which means they don't code for the proteins, and those regions are called introns. Okay, they're called introns. So and there are some regions called axons. Axons are the protein coding regions, and open reading frames are the ones which which code for the for a specific protein. So they have start codons, they have stop codons. So all these things we will be talking in future also. So you can read these concepts from those papers which I posted. So gene annotation, basically there are two kinds, structural and the functional. Structure means to find all these regions on DNA. So if you have a list of ATCGs, where is the promoter, where is the enhancer, which region is the axon, which region is the intron, and where is the open reading frame. So to find all these things is called the finding the structural gene annotation, to do the structural gene annotation. So after the structural gene annotation is done, next thing is to find the functional annotation, to do the functional annotation, which means if you have a gene which codes for some protein, what function that protein plays, that is called the functional annotation. So once the gene has been identified and its axon intron structures determined, the next step is to figure out what's the function of the gene. Functional annotation is usually done through sequence comparisons to protein sequence databases or protein domain motif databases. So functional annotations can be done if you know what kind of protein your gene is going to make. You can do the comparison of the your protein with the other proteins which are present in the database, and that can be done by knowing by the protein domain or motifs. So domains and motifs, they are regions in the proteins. We will talk about domains and motifs in this course as we will be doing the proteomics lectures. So just for now, domain and proteins, they are some regions in proteins which do some special function. And exons and introns, introns is basically junk DNA, they don't code for any protein, and exons are the ones which code for the proteins. So gene annotation is extraction, definition and interpretation of features on the genome sequence derived by integrating computational tools and biological knowledge. Gene annotation includes finding where is the promoter, where does the transcription starts, where is the start codon, where is the exon, where is the intron, where is the transcription termination sequence. This is all the gene annotations. So that's all for this lecture. If you have anything not clear, you can always talk to me. And okay, thanks.